I want to go through this and not operator because it involves a kind of inhibitory input called shunting inhibition. And shunting inhibition may potentially play an important role in many different neuronal signal processing operations, including the construction of this and not operator. So like before, I posed a really trivial, simple little model of an and operator. Well, here's one for an and not operator. So we've got a dendritic cable. It has two distinct inputs. One is an excitatory synaptic conductance change. The other is inhibitory synaptic conductance change. And they have their associated reversal potentials. So shunting inhibitory input is a special kind of inhibitory input. In general, if a neuron has a resting potential of some value E, that is to say, the membrane is permeable at rest to ionic species whose reversal potential is E millivolts, shunting inhibitory input is one in which the reversal potential of the ions that move through the inhibitory conductance change, the inhibitory channels, is also E. Here I'm calling it E sub I for inhibitory. But this is to say that if we activate the shunting inhibitory input by itself, it produces no response because it's increasing membrane conductance to ions whose reversal potential are the resting potential of the cell. And so here in this simple model, we mimic that situation by saying this reversal potential for the inhibitory ionic species is 0 millivolts, so that when this conductance change occurs, it doesn't produce a change in membrane potential if this cell is at rest, because the cell is resting at 0 millivolts in this simple model, or whatever the reversal potential happens to be. So inhibitory inputs produce no response by themselves, but they influence the response to other inputs. And Koch and Peugeot were interested in how they influence response to excitatory inputs. So we can understand this mathematically and we can understand it intuitively. So let's do the intuitive explanation first. It's really simple. Mathematically, we'll do a derivation that is in the steady state. We won't consider transient responses. So in this situation, let's assume that the reversal potential of the excitatory ionic species is 20 millivolts relative to ground. Okay, so that when this synaptic conductant, when an impulse is conducted down the axon projecting to this neuron, a neurotransmitter release occurs, and that neurotransmitter binds at a receptor in this cell's membrane to cause a synaptic conductance change. That membrane conductance is increasing to ionic species whose reversal potential is 20 millivolts depolarized. So that will produce a membrane that'll produce a positive inward current into the dendrite at this point and a voltage depolarization at this point. And if there's no inhibitory input, this will produce some voltage depolarization that will propagate passively to the end of the cable. Let's say the cell body, the soma, is sitting out here, so this is where spike generation occurs. So maybe that voltage is enough to cause spike generation in this neuron. Maybe it isn't, but in any case, some level of depolarization is produced out here. Now imagine what happens if we turn on the inhibitory synaptic event. If an impulse propagates down the axon of whatever neuron is projecting right here and increases the synaptic conductance to an ionic species with a reversal potential of 0 millivolts. Well, as I said before, this event in and of itself would produce no depolarization at the soma, but consider what increasing that membrane conductance does to charge that is being injected into the neuron from this excitatory input and it's propagating to the soma. This is called shunting inhibition for a reason. It's because it shunts that charge out through the membrane. So if this were an infinitely high conductance. So that node would be tied, this node would be tied to ground, so the potential there would be 0 millivolts. Whatever current is injected out here at the end of this cable never gets into this cable, never excites that cable at the end of the cable to be depolarized, never produces an action potential in the target neuron. And so the effect of opening this membrane conductance is to divert charge away from flowing into that cable towards the soma and shunt it out through the membrane. So whatever voltage would be produced by this alone, by adding in this shunting inhibition, that diverts charge away from the soma and reduces the effect that this input has on the soma. So that's pretty clear intuitively. So how do we get an and not operator out of this? Well, this synaptic input has an influence only on the soma, only if it is on and this input is not on. That's the sense in which this neuronal circuit can be an and not operator. If this excitatory input is on and this inhibitory input is on, charge gets shunted away from the soma, the neuron might fire. But this is on, if this one is on and this one isn't on, so and not, then whatever charge is injected into the dendrite here may be able to propagate to the soma and cause the neuron to fire. So this synaptic input ablates that synaptic input, so to speak. That's how the and not, if A is on and B is not on, and not. That's how it comes about. So that's the intuition. And okay, so I think these slides in the handout I gave on Friday are at the very end. I've moved them up. Let's just walk through how we show this without going into the equations. Let's just go through the logic of how we show this. So the first thing we want to show is what is the voltage at this location at the end of the cable when we activate this excitatory input alone. Assume that this inhibitory input is not present. So that conductance is 0. It's not even there. So we know how to solve for the voltage at the end of this cable given this current input is injected into the other end of the cable. We've got the equations for that, and we can work it out. So we won't do it. This is how you do it. It's a simple problem. You know what the solution is. I presented it the first day. And so that's our kind of baseline voltage. We want to ask the question, when we make this synaptic input non-zero, when that conductance is non-zero, how does it attenuate, how does it reduce this voltage? When we deliver both excitatory and inhibitory input, how is the voltage at the end of the cable at the soma reduced relative to this voltage? Okay, and we'll refer to that reduction as the attenuation factor. So the next step here is to ask the question, okay, well, what if we ignore the excitatory input and only apply an inhibitory input? So this synaptic conductance is now non-zero. It's something other than 0. So again, whatever that synaptic conductance is, whatever this reversal potential is, we can solve that problem. Because we know from the formulas that you've seen before what the input conductance of this branch is and what the input conductance of this branch is. Therefore, we've got the input conductance to this cable structure looking out of that node. And therefore, with this current and that input conductance, we can compute voltage at that node. We've got it. And then we can compute how that voltage declines along this cable to where the soma is located. So that's for an arbitrary inhibitory reversal potential. But for shunting inhibition, I said that reversal potential is 0. So we don't need to go through all of that. We know that if this battery is at 0 millivolts and this is tied to ground, changing this conductance doesn't inject any charge into this cable structure. So there's no voltage out here. It's 0. It's easy. Okay, that's the easy case. Now what if we apply them together? Well, so we'll start back at this end. So both GE and GI are applied together, which means they're both non-zero simultaneously. There's both excitation and inhibition. So we know in general what the strategy is for solving cable problems for voltage along a cable. We first have to work out what is the terminating conductance of this little chunk of the cable. If we can figure out that terminating conductance, then given a current flow into one end, we can compute the voltage produced at that end and how it decays along the cable to its endpoint. So we know what that terminating conductance is. It's just from sitting at this point on this cable and looking outward, the terminating conductance I see is this conductance in parallel with this input conductance. We've got that value, right? And we can compute this value. Just take the length of that chunk of cable and determine its input conductance, assuming a sealed end boundary condition, which is what I've assumed here. And so we know how this cable is terminated, and therefore we can get voltage everywhere along that cable in response just to this value GE when GI is something other than 0. So the effect of this inhibitory conductance is only to change the terminating conductance on that cable, right? It doesn't result in any charge flowing through this pathway due to an electrical driving force other than the voltage gradient between whatever the value is at this node and this battery. Okay. So if we calculate voltage at this node, then all we need to know is the input conductance of this cable to get the voltage at that end node. And so it's this mess here. That's why I don't want to go through it, because it's just a foolish mess of hyperbolic functions. But the neat thing that Will Rawl did, as you know, is he worked out the theory for calculating these things on pencil and paper. So here it is on pencil and paper. This is the voltage at that location when we turn on both of these conductances. It's this stuff here. And this was the voltage at this location when we turned on only this conductance. And so the attenuation factor we want is the ratio of this voltage when both are on to this voltage when only the excitatory input is on. That ratio will tell us by how much does the presence of this inhibitory synaptic conductance change reduce the voltage along the cable. And so here is a plot of that voltage. And to get this plot, I have to make some assumptions about the values of the inhibitory synaptic conductance and the excitatory synaptic conductance. And so for simplicity, I'm plotting the inhibitory conductance normalized in units of G infinity, which is the input conductance of the semi-infinite version of that cable. So that's what's plotted along this axis. And I've just arbitrarily picked a value for the excitatory conductance to be 10 to the first power. And so as we increase the inhibitory conductance, this curve here shows by how much we're attenuating this voltage ratio. So if the inhibitory conductance is 10 times larger than the excitatory conductance, it's substantially reduced by a factor of 10, that voltage. So it's essentially shunting charge out through the membrane, preventing any charge from getting to the end of the cable, attenuating the voltage at the end of the cable. It's functioning as an and not operator. Now in the real world, synaptic inputs are transient, right? They have a certain duration. They're applied for following an action potential that causes neurotransmitter release, these conductances will change for fixed amounts of time. And so there is another factor involved in the and-not operator. If this synaptic conductance as activated by neurotransmitter release, let's say it has a duration of 5 milliseconds, so it opens, charge is injected in, that charge will begin to flow down this dendritic cable. It will take some time for that bolus of charge to reach this node. There's a propagation delay. And if we then turn on this inhibitory conductance at about the time that charge is moving past here, then this inhibitory conductance can effectively shunt that excitatory input and negate it. But if we miss that timing and this inhibitory conductance change is late, that wave of charge will have already passed this point and be propagating towards the end of the cable. And so this inhibitory conductance change will be much less effective. And so in real neural systems, there are some timing considerations. The inhibitory input has to occur at about the right time when charge is moving by that point to shunt that charge out through the membrane. And if we were to reverse the positions of the excitatory and inhibitory input here, suppose this was the excitatory one, this was the inhibitory one, well, this shunting inhibition has the property that it's only effective if it's placed on the so-called direct path that charge must take in flowing towards the site of excitation. So if this were the site of inhibition, when positioned in this way, that inhibitory input would be relatively ineffective at vetoing the actions of excitatory input applied closer to the end of the cable, closer to where we're modeling the soma or thinking of the soma here. So there's this also, so there's timing that's important, and there's this on-path condition. The inhibitory input must be on the path that charge from the excitatory input takes as it's propagating towards the site of neuronal excitation. But if those conditions are met, then we see from that curve that I showed you that inhibitory input can very effectively veto excitatory input, and that's how the and-not operator arises. And so Koch and Poggio enjoyed playing these games of hypothesizing patterns of excitatory and inhibitory input and imagining the kind of logical operators that they could compose by doing this. So here is an example of a logical operator that could be constructed by positioning excitatory and inhibitory inputs in this fashion on the dendrite of a neuron. It's this logical operator here. I don't know the value of this logical operator, but they were showing how composition of logical operators can occur in neuronal dendrites. So what does this mean? First of all, E3 and not I1 or I2 or I3. That means that E3 affects what happens at the soma, affects neural firing if it's on and I1, I2, and I3, the inhibitory inputs are not on. So that's part of the logical operation implemented by this pattern of inputs. Or it could be that E2 is on and I1 and I2 are not. Remember, I1 and I2 are, in this drawing, on the direct path that charge must take when propagating from the site of charge injection at E2. So that charge propagates to the soma. Since they're on the direct path, they can effectively veto charge from E2. But I3 is not on the direct path. It's behind E2. So I3 doesn't appear in this relationship. It's not effective at vetoing the charge that flows towards the soma, so it doesn't appear here, right? And then, of course, there's the remaining excitatory input, so we have or E1 and not I1. So This all together is the logical operation that this pattern of inputs might perform. And so it is in this sense that Koch and Poggio meant that neuronal dendrites are capable of performing universal computation depending on the relative placement and, of course, strengths of excitatory and inhibitory inputs. So you know the conditions on the relative placement. Inhibition must be on the direct path to implement an and not. And the relative strengths, you've gotten a sense from that last slide that the inhibition has to be fairly large relative to the excitation for it to do the shunting. But if those conditions are met, then in principle it's possible for neurons to implement these kinds of more complicated logical operators on their input. And we can imagine other scenarios where the AND operation, suppose there was another a dendrite coming off this neuron, and this set of inputs was duplicated on that other dendrite, then we'd have all of this mess and all of the other, and this pattern replicated in the other dendrite. So, right? So we could imagine a scenario where one of these signals must get through, and another set of complementary signals on the other dendrite need to get through to cause this neuron to fire. So in other words, by adding a dendrite, we might be implementing an AND detector on these two distinct sets of inputs in the dendritic tree. So that's the way in which these neurons can compose logical operators. And Koch and Poggio went on to argue that this may be the basis of directional selective responses in the retina. So they hypothesized a retinal ganglion cell which may be sensitive to movement of light. I should say this is the null direction, insensitive to movement of light in this direction, but a movement of light across the receptive field in the other direction would excite this neuron. And so the idea of how the null direction, oh yes?
On the last slide, why are there the ors in between like the terms?
Yeah.
So, like if you have multiple dendrites, they all add up at the soma, but like if you have multiple signals on one dendrite, wouldn't they add up?
Yeah, so what the OR is saying that this could happen. That's one way the neuron can fire, is if there's an excitatory input here and these inhibitory inputs aren't on. But there's another way for the neuron to fire, and that is if there's an excitatory input here and these two inhibitory inputs are not on. That's this condition, and therefore the neuron can fire if this condition is true, or if this condition is true, or if this condition is true. This excitatory input is on and this inhibitory input is not activated. That's where the ORs come from, because there's multiple ways that signals from excitatory inputs could get through on this one dendrite. These are all the ways in which those excitatory inputs can get through to cause this neuron to fire. And then imagine with the if there were another dendrite, that we were to just shift all of these out, and so there'd be some minimum distance. We'd be asking the same question that I'm asking in the homework. Where do the input, what is the distance L from the soma that supports the creation of an AND operator? So if we were to push this family of inputs out on the dendritic tree, that's L, that distance. And on the other dendrite, push them out on the dendritic tree for that same distance. We'd be composing a logical operator, which is this and the same kind of logical function with the other set of inputs on the other dendrite, if that makes sense. That's how we can build, we could throw an AND operator into the works along with all of this stuff. It's just an example of how complex logical operations can be composed in dendritic trees. Okay, the null direction this is a hypothesis, comes about if this neuron that is impinging onto the retinal ganglion cell, if this is considered the filled black circles are excitatory inputs and these black rectangles here are inhibitory inputs, and if the timing is right, the velocity of the bar is right, the travel time of the signal to the site of inhibition is correct, if all of these things are adjusted correctly, then the neuron is excited by this synaptic event here. The light bar moves slowly over to this location, exciting the presynaptic neuron. It excites the postsynaptic neuron, but the inhibition gets there before that wave of excitation does. So the inhibition can then veto this response, and so on and so forth. So the light bar moving in this direction results in a pattern of lateral inhibition that prevents the charge that is injected by these excitatory synaptic events from getting through and reaching the soma. So they put this forward as an example of one way in which this kind of patterning of inputs and patterning of excitatory versus inhibitory inputs could contribute to receptive field properties of real neurons. Okay, so that's a summary. So far we talked about dendritic integration, how taking some net synaptic conductance and spreading it out across a dendritic tree is a more effective way of driving a neuron than lumping all that synaptic conductance change together at one megasynapse within the dendrite. So dendritic distribution is a good thing. Sending inputs to electrically distinct regions of the cells is good if you want to effectively drive and excite the target cell. And then we talked about building these neuronal and not operators and how neurons can perform logical computations of arbitrary complexity in the dendritic tree. The final topic, okay, is dendritic spines and ideas about the role they play in information processing in the nervous system. So, have you run across the name Ramon y Cajal yet? Have you seen? Okay, very famous Spanish neuroanatomist. There you go. La textura del sistema nerviosa del hombre y los vertebrados. I'm sure I'm doing a terrible job at pronouncing that, but he wrote this very famous neuroanatomy textbook jam-packed with hand-drawn diagrams of structures that he saw in the nervous system under the light microscope. This was in 1899. So he was the first person to notice these little appendages that come off dendrites that are called dendritic spines, and they have a characteristic shape. They have a shaft, that's the shaft, and they have a head, that's the head. Shaft is very, very low diameter, very small diameter structures. We'll see the diameters in just a minute. He discovered those, and so many cortical neurons are just, the dendrites are absolutely covered with these spines. And that's where synapses land. They land on the heads of spines, predominantly. That's where synaptic inputs are made. My pictures of synaptic conductance changes being applied to a dendritic cable, at least in cortex, that's really not an accurate picture. Synaptic inputs land on these spine heads, and that's where the synaptic conductance change occurs. And so whatever charge flows into these spine heads has to flow through these narrow spine shafts to get into the dendrite to influence what the neuron does. Okay, what's the intuition here? As always, right? So these spine shafts are very, very low diameter. In some way they must limit the amount of charge you can inject into a dendrite. And they do. They're small enough, fine enough that they limit the movement of charge into a dendrite. And if you can regulate those dimensions, then you could regulate synaptic weight in the nervous system. And so it's interesting that here's a way of adjusting synaptic weight that in a way kind of sacrifices the effectiveness of a synapse. You can do it by pretty much attenuating the amount of charge that reaches the dendrite. And so being able to adjust synaptic weight, even if it means attenuation, may be an important design principle in the nervous system. And of course, we've got much higher resolution reconstructions of spines. So here are some examples. Here's some super high resolution drawings of spines. I think these were, so these are going to be either 2-photon imaging reconstructions of spines, and or they might be electron micrographs. I'm not sure which they are. But you can check it out on Synapse Web and see what they are. So here's another example of a dendrite covered with this mess of spines. It's just amazing. So here are some dimensions. The width of the spine neck, that's that thing I called a shaft, they're calling it the neck here. The neck width, 0.09 to 0.5 microns. Oh my, that's small. The neck length, the shaft length, 0.1 to 2 microns. That's pretty small too. These are very, very small structures. That's the bottom line. Oh, it's interesting. Look at this. This is super high resolution imaging done in live tissue, not in fixed tissue. That's pretty neat. You can image these very fine structures in living neurons. And that led to an interesting finding, in fact. Christoph Koch was a good friend with Francis Crick of Watson and Crick fame. And in his later years, Francis Crick became he really went into different areas of neuroscience, and he became interested in neuronal signal processing. And they were at a conference, at a retreat somewhere, and sitting around a campfire. There's a paper that has this story in it. And Francis Crick began speculating, what if spines could twitch? What if they could move? Because actin filaments had just been discovered in spines. And you know, actin is monomeric or polymeric. It polymerizes to form strands of actin monomers. And actin had just been found in the shaft of dendritic spines. And so Francis Crick was speculating, what if these actin polymers could assemble and disassemble and thereby control the length of a spine, that spine shaft? And he thought that would be a way of controlling synaptic weight. So Christoph Koch heard this and scrambled off, and with Tommaso Poggio began analyzing some of this. But here is some other interesting motivating data, again from living neurons, where they've got the spine membrane fluorescently labeled, and they're measuring the length of spines as a function of time, and it's changing as a function of time. So here's 0 to 18 minutes. This spine that they were imaging started off with a length of 2 microns, and it went up to about 3 microns. Here's a spine that over the course of half an hour, its length went from 0.5 up to a peak of 1.5, and then it dropped again, and it went up again. And here's a spine that just popped out of nowhere. It just emerged from the dendrite itself. And so all of a sudden we've got a spine that emerges, and it has a length, the shaft has a length of about 1.5 microns, and now it goes away again. And so this was actually a visualization of dynamic spine creation, yeah.
Does the transient spine occur at a place that already has a synapse, or does it just kind of happen due to some other cause?
I don't really know the answer to that. I'm not sure. Sorry. I don't know. I'd be speculating if I said any more, so I don't know. It's a good question. So here's Christophe Koch and Tommaso Poggio's first shot at thinking about dendritic spines, and it's this kind of electrical circuit model. And so this part of the model represents the shaft of the spine, and this represents the head of the spine. And the circuit diagram has some unusual things about it that we need to talk to, and some assumptions that are buried in it. So the first kind of unusual thing when you see this kind of circuit diagram is, why is ground sitting here and here? Well, that is because we take as ground the extracellular space. So ground is representing the extracellular environment outside of this dendrite. And this spine has a shaft that's protruding from the dendrite, an enlarged spine head, but on the other side of the membrane forming that spine head is the extracellular space. So that's ground 2. And the assumption is that these two locations are isopotential, they share ground, it's ground, extracellular space is just the ground. So that's why there are these two symbols for electrical ground in this electrical circuit diagram. So that's part 1. So let's first talk about the spine head. So if this is ground, this is outside the spine head, this is inside the spine head, this is the voltage in the spine head. And whatever the surface area of that spine is, a synapse landing on the spine causes some synaptic conductance change, and those receptors, whatever they are, become permeable to ions with a reversal potential shown here. So this is the effect of a synapse, of injecting charge into the spine head. There's a core resistance of the spine head, and there's a capacitance of the spine head. And we're ignoring both. We're going to do a steady state analysis, and we're going to assume that the surface area of the spine head is so small that whatever other channels are in it, they contribute negligible conductance. And for that matter, the surface area of the spine is so small, it has very little capacitance. So we'd argue that both of these pathways don't actually exist. And so the major permeability in the spine head membrane is when there's a synaptic input to the spine head and membrane conductance changes in response to that synaptic input, generating an inwardly directed synaptic current. Okay. Okay, this represents the spine neck. Now, if you recall the pictures of the spine neck, it's like a little tiny cable, but we're not modeling it as a cable because it's so short. So we forget the cable properties of this little tiny cable. It's about a micron long or so, and it's so short that we're going to assume everywhere along that cable is isopotential. We're just going to represent the neck using a resistor, not a cable-like representation, which would be a core conductance with parallel RC current pathways coming off that. That's the cable representation, right? So we're not going to do that. We're just going to represent it as a resistor, and that spine is connected to the dendrite itself, and so the current through the spine head gets directed into that dendrite, and looking into that dendrite, we call this the input resistance of the dendrite where that spine is located. Okay, that's the setup. So, Ohm's law gives us the synaptic current. It's just the electrical driving force, the potential difference across this resistor times that conductance, so that's that current. The voltage at the spine head is due to the voltage drop produced by the synaptic current flowing through the resistance of the spine shaft, I'm calling it R sub n for the spine neck actually here, which is in series with the input resistance of the dendrite. So the voltage at the spine head is simply this voltage. And if we lump this spine neck resistance together with the input resistance of the dendrite and call that R, then we can plug that into this equation and just get voltage at the spine head in terms of the reversal potential of the ions that are moving through the synaptic conductance, and a term that depends on the synaptic conductance times this net resistance. The voltage at the dendrite is in turn equal to so the synaptic current is flowing like so. It can't flow, remember these are zero, so it can't flow anywhere else in this electric circuit. It has to flow through this resistor. It has to flow into the dendrite. There's no other place for it to go because the dimensions of the spine are so small. So the voltage at the dendrite is just the synaptic current times the input conductance of the dendrite itself. So that's the synaptic current, and that's the input conductance of the dendrite. So that can be simplified. And if we now take the ratio of the voltage at the dendrite to the voltage at the spine head, we get the drop in voltage that is due to current having to flow through the spine head, through the synaptic conductance, and into the dendrite. So this is the ratio of voltage at the dendrite to that at the spine head. And so the question is, what are the factors regulating that? So if we divide this term by this term, it comes out to be equal to the input resistance of the dendrite over the sum of these resistances. And then if we do treat, if we define the resistance of the spine neck as being governed by the diameter of the neck and the length of that tube and the core resistance of that tube, it's like thinking of the spine neck as a single little compartment. We can write that resistance in terms of the length of the spine neck. And so obviously manipulating the length of the spine neck controls this resistance, and in that manner how much charge gets into, or the voltage that is produced at the dendrite in response to a synaptic input at the spine head. So even though I'm using little l here, it's the same L as the capital L on the previous slide. So here I'm varying spine length and for a spine of some particular dimensions, which I took from that table that comes from the real anatomy. And I'm plotting how that controls the ratio of voltage at the dendrite to voltage at the spine head. So obviously varying spine length is a way in which you can regulate the degree to which you're regulating the voltage that a synaptic input to that spine produces at the dendrite. So if your baseline length of the spine neck is 1 micron, you make it shorter, you amplify that voltage. If you make it longer, you can attenuate that voltage. Yeah.
You said that the actin fibers were changing the length of the spine, but what's telling which actin fibers the length of their contraction?
Oh, nobody knows. It's not known. It isn't known. And in fact, this may not really be the way that synaptic weight is truly regulated. It is the case that spine length is adjusting, and lots and lots and lots of ATP levels will control that. Calcium levels control that. It's one possible mechanism by which, where there are physiologic variables and principles that could govern the length of the spine. But it is more likely, I think, that synaptic weight is probably regulated by other things more importantly. And they were complicated to get into, so I decided not to talk about them. But calcium levels in the spine head control the rate at which receptors, synaptic neurotransmitter receptors are implanted into the spine head membrane and controls the rate at which they're removed from that membrane. So if you place more receptors in the spine head, that's a way of amplifying the response to a synaptic event, because you've got more receptors that open and inject more charge into the spine head. And it's known that calcium can also regulate the conductance states of those ligand-gated receptors. So when they're phosphorylated, they actually conduct more charge than when they are not. So calcium regulates both of those quantities, and there's a beautiful model that looks at both these processes independently and shows how they can both be reduced and represented using the same modeling formalism. And so it doesn't matter in a way in which mechanism is true. They both have the same mathematical embodiment and can be studied using the same kind of dynamical equations. And in that paper, these investigators show that, in fact, with these kinds of assumptions, via calcium regulation, it's possible to also regulate synaptic weight. So there's multiple mechanisms by which the nervous system can use feedback to the spines. I should say that an important signal in doing this is voltage. A signal is needed to reinforce, the idea is that reinforce synaptic weight when the neuron fires. Well, how the heck does a spine know that the neuron fires? Well, we now know, and it can be shown from simple models like this, that whatever voltage, neuron fires an action potential, that large depolarization is not just going to propagate down the axon of that neuron if it has one. It's going to propagate back out into the dendrite. In everything we've said, we've thought about how does charge get funneled from dendrites to soma to cause neurons to fire. Well, dendrites can sense when neurons fire because they sense a big voltage wave that comes back out into the dendrites. And because the spine head resistance is so high, there's very little voltage drop from this point back up into the spine head. So spines are structures that can very accurately sense the voltage at their dendritic location. And it's believed that that signal, together with a calcium signal, helps reinforce synaptic weight, that both mechanisms are at play here. And so a neuron has a way of knowing what signals cause the neuron to fire, and there's a calcium and voltage dependent mechanism by which that gets signals back to the spine heads. And through these voltage dependent calcium mechanisms can increase synaptic weight in an automatic way to reinforce synapses that are causing the neuron to fire and to reduce the effect of other inputs that don't cause the neuron to fire. So that's called Hebbian learning. And there now is a molecular basis and mathematical models of Hebbian learning that are quite neat. So neurons are capable of obviously, we're all fairly conscious beings of remarkable computations. And some of the key figures have been Will Rall and Christoph Koch and Tommaso Poggio and John Rinzel and Idan Segev, whose name I didn't mention, but he wrote that paper. I showed you that paper that's 60 neurons from human cortex reconstructed. That was work by Idan Segev. These have been the leaders in real neural modeling of dendritic trees and signal processing in dendritic trees. It's a neat story. I'll end there. Did you have a question? Oh, okay, sorry. All right, thanks.